Have you ever been told that transcription must occur in the 5-line to 3-line direction? This topic can be very challenging to understand if you are not paying close attention. First, imagine if you have 2 strands, but then you need to make one more strand from those last strands. What would you do? Well, want to know more? Let us tell you.
First, let's understand the RNA structure. RNA nitrogenous bases are almost the same as the DNA bases. RNA has guanine, cytosine, and adenine. However, different from the DNA, RNA doesn't have thymine. Instead of thymine, RNA has uracil. The guanine pairs with the cytosine and the adenine pairs with uracil. All these nitrogenous bases are joined together by a strand made of phosphate group and ribose, a sugar. Also, in this process, we are going to see some enzymes, which are proteins that help speed up chemical reactions in our body.
Transcription is the first step in the process of using the genetic code in DNA to synthesize the proteins in our body. RNA transcription starts inside the nucleus, but when it ends, it goes to the roof endoplasmic reticulum.
Transcription begins when the enzyme RNA polymerase attaches to a segment of DNA. Then, a particular area of the DNA helix called the promoter is unwound by the enzyme RNA polymerase, separating into 2 strands.
Now that the double helix is open, we ended up with 2 strands of DNA. One is the template strand and the other is the non-template strand. First of all, we need to understand that the transcription is built in the direction 5-line to 3-line. However, the direction of the template strand will be the opposite, 3-line to 5-line. I know that may seem hard to understand for now, but let's check it out. So remember, the leading strand in this process is the one that is 3-line to 5-line, and the non-leading strand is the one that goes 5-line to 3-line.
Wow, that was the hardest part to understand, I promise. Now that you know which strand will be the template, we can start saying that the enzyme DNA polymerase attaches to the template strand and, using this strand as a guide, completes the nitrogenous bases that you saw in the beginning of the video throughout the entire strand.
As the strand is being transcribed, the new strand can be 3 different types of RNA. They are transport RNA, tRNA, responsible for linking codons and amino acids, ribosomal RNA, rRNA, the main ingredient to make ribosomes, or messenger RNA, mRNA, responsible for transporting the information transcribed from the DNA chain.
And with that, when translation ends, the RNA leaves the DNA and goes to whatever place it needs to go, finalizing with DNA closing again. Transcription is essential because it transforms DNA information into RNA, allowing the production of proteins and the proper function of the cells. This process is vital to life, and errors in it can cause illness.
Quiz time! What nitrogenous base does RNA have that DNA does not?
What enzyme attaches to the DNA to begin transcription?
Which direction must transcription occur in?
Which strand is used as the template during transcription?
Which enzyme completes the nitrogenous bases during transcription?